Uh, yeah, so good afternoon, everyone. Today, I'm really excited to talk about our efficient generative model to study the mitochondrial morphology. So our motivation comes from a recently really popular topic, which is the object-wise evaluation of subcellular organelles. With the development and progress with latest technologies, including the focused ion beam scanning electron microscope, which is the fifth sim, enable us to resolve the individual organelles objects and fully capture their shapes. For example, for the image in the right, the fifth image of the mitochondria allow us to see clearly of the, see the boundary and shapes of the mitochondria clearly. However, previously, such accessibility is not available. For example, for the image on the left, the confocal microscope image just can only be able to capture the rough spatial distribution of the mitochondria in a cell level, where the mitochondria is tagged with green biomarker. So with such accessibility of individual subcellular organelles shapes, it also enables some innovative quantitative study of the shapes of individual organelles, which will help us to understand some really complex biological process. For example, the mitochondria, which is correlated with several important cell functions. um, we know that right before the autumn, right before the mitophage, the shapes of the mitochondria will vary from the others, which release IGMI signal to resulting in the engulfment of the autoprotomic, uh, autoprotomics. Um, And such study will demand an efficient generative model to estimate the shape distribution of the organelles population. And here's why we want to propose and design such a model. So the first step of our model is to portray the mitochondria in a three-dimensional space using spherical harmonic transform. We know that the most available image of mitochondria for a whole cell is contained in the three-dimensional TIFF image, which is essentially a stack of 2D slides. Uh, we can see it from the leftmost image. The white in the image means the space inside the mitochondria, while the black means that the space outside, so that we can further apply spherical harmonic transform SHT to obtain the Sform coefficients. So you may curious about what do I mean by spherical harmonic transform? Well, it is a transform that is similar to Fourier transformation, which illustrates that a sequence of complex signal can be considered as a linear combination of uh, several simple trigonometric bases, including sine or cosine functions. Similarly, in SHT, we will first map the original shape into a spherical unit, which is the R here. It is the spherical coordinate. Then by applying the spherical harmonic transform, we'll be able to represent the spherical coordinate with the linear combination of the spherical harmonic bases, which are y here. And we will be able to obtain the coefficients, which are c here. And the c can be later applied as a descriptor, based on which we can perform the inverse transform to reconstruct the shape. So you may wonder what I mean by descriptor. Well, I would take a vector v in the three-dimensional space as an example. We know the three-dimensional space is spanned by, the three-dimensional real space is spanned by three unit vectors, u1, u2, and u3. And we can see that the v can be represented by a linear combination of these bases. And so 3, 2, 5 can be act as a descriptor of v in the three-dimensional real space. Similarly, we will be able to apply this. Uh, for SHT, we will be able to use the SHT coefficient as a descriptor to describe our shape. Yep, and the second step is to apply the the principal component analysis to reduce the dimensionality for further shape reconstruction, because we noticed that the SHT coefficient is a really high dimension vector, and we certainly do not want this. So while choosing a proper K, we are expect that these principal components can cover more than 75% of the total variance, which is similar as the threshold in Rafalski's work, which is published in Nature in 2023. So we are really happy to notice that for the first 5 principal components have already covered more than 75% of the total experiment variance. However, in our numerical experiment, we did the number of principal components from 6 to 10. And our final step is to perform the shape reconstruction. We adopt the process again from Rafalski's work that they also did a similar process in their work. The base process looks like this. The first step is to obtain the um, standard deviation of corresponding principal component. And then we obtain this critical point and map to the SHT coefficient space by applying the inverse PCA transform. Um, Based on the coefficients, we can use some open source rendering software to visualize the shapes. And in their work, they also observed the reconstruct shapes from 3 views orthogonally. Actually, we also did the same thing. We just first apply the PCA inverse of K principal components. So basically here, what we did is that I would take the first image, first mesh in the first row as an example. So what we did is like we take the principal component 1 to be -2 sigma, where sigma is a corresponding standard deviation of the first principal component. And we keep all other principal to be 0. And we just like map this point to back to the 1024 SHT coefficient space. And finally, we just like render the reconstruct shape by open source software like Cell Organizer. And we also observe it from the first view. Remember that the first view is the side view 1 from Rafalski's work. So what we did in our experiment is we plot first 10 principal components for a set of cells a set of cell lines, including the macrophage, the HeLa, and and the JERCAT. The the percentage under each name of the principal component is their corresponding experiment variance. So from the graph, we can see that we can only see the noticeable variation in the shape of the in a reconstruct shape for the several top principal components, like the top 5 or top 6 principal components. because they have a relatively large explain variance for the, I mean, for the tailing principal components, we might not be able to observe noticeable variation in their shape. And we're also curious about whether we can just like distinguish different cell lines just depending on the shapes of their mitochondria. But it seems like, I mean, from this image, it seems like the mitochondria obtained by, I mean, reconstruct by our model are not distinguishable from each other, so that we can just like draw a short conclusion that we may need more cell lines or more or a larger data set to perform such analysis. Hopefully, we can see some significant result in the future. Yep, and it is also what I want to talk about in the discussion part. It is illustrate that our generative model can help the researcher uh, to perform quantitative study of the shape for individual mitochondria based on a general population of the certain cell lines. However, as I discussed previously, large sample size and more or data from more cell lines might be in demand, because currently we are only able to utilize some public data sets that are, I mean, that only contains thousands of samples. I mean, thousands of samples and just like 5 or 6 cell lines. And I also want to point out that another approach to further study the object-wise evaluation of subcellular organelles is to perform the spatial distribution of various organelles. For example, we may also curious about the distribution of the distance for the between the mitochondria uh, and other organelles like the lysosomes or the cores of the cells or the boundaries of the cells. And here is all what I would like to talk about it today. Thank you.
Any questions?
Yeah, quick question. Just wondering if the variation in the shape have any biological content,
Yeah.
whether that affects the function of mitochondria.
Sorry, could you please repeat again?
Again, the morphological variation, whether that affects its function of mitochondria.
The morphology will affect the function
The morphology of the
mitochondria. Would it affect the function of the mitochondria?
So you mean the scanning of, I mean, you mean the process to obtain the data set, or you mean the analysis to?
Copy the shape. Why is the shape of the mitochondria? What is the effect of the shape is different?
You, like it will depends on the coefficients we obtain during the SHT, because some coefficients will I mean some coefficient will include the information of the rotation of the mitochondria, and some will correlate it with its shape, like it's large or small.
Right, but if it's large or small or different shape, what
Yeah.
does that mean for the biology? What does that change the
Yeah,
biology of the cell?
that's a really good point. I mean, that's probably, I mean, I'm so sorry. I mean, that's probably a future direction to study further. I guess, I mean, at the very beginning, we just guess that maybe the HeLa will have a relatively larger mitochondria, because some publication indicate that I mean, for the cancer cell, the mitochondria tend to be larger, but we just like did not observe this in our study. I'm not sure whether it answer your question because it's like, yeah, it really has some biological insight. Like I mean, sometimes the size of the mitochondria will contain some biological insight, which we are really curious about, and we really want to figure them out. Yeah, and it's one future like direction.
Certainly in yeast, there's mitochondrial shape and size are linked to specific kinds of mutations. We know that. So you may find similar things in other species as well.
Oh, yeah. Thank you.
Was there a very brief question?
Very brief, but sorry if I missed this. Are you segmenting the individual mitochondria in the beginning? That's your first step.
Oh, yeah, that's it. Actually, we just like we just apply some existing segmentation algorithm that are provided by the dataset. Actually, the dataset provides some segmentation algorithm to like help us directly segment each individual organism.
A quick follow-up question from that is, why do you think that you don't see so much variation in shape in PC2? PC2 captures like 7% variation, but even if you go plus or minus 2 standard deviations, it doesn't look like it changes all that much. So what is PC2 capturing?
That's a really good question. I would say that maybe because I mean PC2 has already have a relatively low XBM variance. You can see that it's like below the 10%. So I would say that maybe most of the information is carried by the PC1. Yeah, that's my.
Okay, let's thank you.